And in this video, we're going to focus on a different kind of cell division called meiosis. Meiosis has one purpose, unlike mitosis, and that purpose is to make sperm and egg cells for sexual reproduction. So we need to define a couple of terms here. Homologous chromosomes are chromosomes with the same genes in a diploid cell, in an adult cell. You have one copy from each parent. So diploid cells have a full set of chromosomes, one from each parent, or we say their number is 2n, 2 copies of each chromosome. Haploid are gametes, sperm and egg cells. They have only a single set of chromosomes, or 1n in number. Let's review mitosis. Remember PMAT, prophase, metaphase, anaphase, telophase, tells us the order, and you have the different steps of mitosis. Meiosis has the same 4 steps, but they happen a little bit different. And there's a few things that happen in meiosis that don't happen in mitosis. And also, the cell's going to go through the stages twice in meiosis. So you have meiosis 1 and meiosis 2. So meiosis is the process in which the number of chromosomes per cell is cut in half through the separation of homologous chromosomes in diploid cells to make haploid cells. And the reason for that is because the function of gametes, the sperm and the egg, is to join in fertilization and become a fertilized egg, a zygote, and so on. If we didn't cut the number of chromosomes in half, every generation would have more chromosomes, and we know that having just an extra copy of one chromosome can cause things like Down syndrome. Having multiple copies of all the chromosomes is almost always lethal. So in prophase 1, the first step of meiosis 1, happens a lot like in mitosis. The cell's going to copy the chromosomes, the spindle's going to form. But one thing that happens that's unique is the crossing over, where you have pieces of the chromosome from mom and pieces of the chromosome from dad crossing over, breaking off, and exchanging places. So now you have genes that were inherited from mom that sort of mixed in with the genes that are inherited from dad on the same chromosome. This helps with creating that genetic variability, which is important for evolution and long-term survival in changing environments. So a tetrad is a group of 4 sister chromatids that forms during prophase 1. Crossing over is where they exchange, like we talked about. Metaphase, the tetrads line up at the cell equator. Spindle then lines them up. Anaphase 1, homologous chromosomes are pulled opposite poles. Telophase 1, then cytokinesis, the cell splits, and each cell, now you have 2 cells that still have 2 copies of each chromosome at the end of meiosis 1. Between meiosis 1 and meiosis 2, there's no DNA replication. The chromosomes do not form tetrads. Instead, it looks a lot more like traditional mitosis, except there is no replication of the chromosome. You can see the stages here, very similar to what we're used to seeing. But instead of separating homologous chromosomes, meiosis 2 separates sister chromatids, parts of a replicated chromosome. So in metaphase 2, the chromosomes are going to line up. In anaphase 2, the paired chromatids separate. So at the end of meiosis, you have 4 daughter cells that are produced that have half the normal number of chromosomes. They're haploid cells. This just kind of summarizes the steps here. You can see this in your textbook as well. So again, if you compare the 2 stages, mitosis produces diploid cells that are identical to the parent cell. There's 2 daughter cells produced. It's asexual reproduction, it's growth, it's repair. The only variability comes if there's a mistaken copy in the DNA, a mutation. Meiosis, instead of 1 round of replication, you have 2 rounds. You have crossing over, you have the random arrangement of the chromosomes at the cell equator that shuffles up the genes and creates genetic variability. At the end, you have 4 daughter cells that are produced that are haploid, and those daughter cells are either going to be sperm or egg cells, depending on if they're produced in males or females. So in this process, in crossing over, we learned that some genes tend to be inherited together because they're located close together on the same chromosome. Things like in humans, hair color and eye color tend to be linked to genes. People with light colored hair often have blue or green eyes. People with dark colored hair often have brown eyes. But it's not always the case because sometimes you can get crossing over happening. The further apart the genes are, the more likely crossing over can happen. And we can use that crossing over frequencies to map the relative distance of genes on a chromosome. It was first done with fruit flies, but it can be done with anything. And this is a map that was created through crossing over of these different colors, these different genes on the fruit fly chromosome. This is chromosome 2 of the fruit fly, for instance.